Okay, it's 4 o'clock in the morning and we're going to approach the Microbial Observatory. This project is coordinated by Dr. Anders Lansen and Dr. Lure Pelde, and hopefully today we will be able to check the effect of soil temperature on gene expression profiles of the soil microbial communities. So it's kind of dark and we still have a long way to go until we reach the Microbial Observatory.
Human life depends on proper conservation of the thin surface layer covering our planet's crust, the soil. In addition to providing food and raw materials, the soil ecosystem also regulates the basic life processes on Earth, like degradation of organic matter, the global cycles of carbon and water, and regulation of the climate. Although it's very difficult to estimate the economic value of the critical services provided by the soil, estimates show that mismanagement of soil biodiversity costs more than $1 trillion every year worldwide. We know that microorganisms play a vital role in this ecosystem, but in spite of this, our understanding of the soil microbial communities is relatively poor. Soil microorganisms include both bacteria, fungi, and small single-cell eukaryotes. Because of their intimate contact with the environment, microorganisms are often more sensitive to changes than larger organisms, like plants and animals. Therefore, it's not surprising that changes in the soil microbial communities often happen before changes in distribution or diversity are observed in the plant communities. Their sensitivity and ecological importance makes soil organisms excellent bioindicators of ecosystem health and changing environmental conditions, like contemporary climate change. Because of this, the Soil Microbial Ecology Group of NEIKER-Tecnalia established a Microbial Observatory of Global Change in the Ordesa Monte Perdido National Park in 2012. Its main objective is to monitor the effects of global change on soil sustainability.
We are at the beginning of the Microbial Observatory. The name is Behatoki, which in Basque it means observatory, but we are interested in the changes in the soil microorganisms, particularly bacteria and fungi. These microorganisms are essential for the functioning of the soil ecosystem and also, of course, for the functioning and survival of the plants. All those trees and plants that we see, they also depend on the many soil functions performed by the soil microorganisms. So as I was saying, we are interested in the changes in those microorganisms that cannot be seen with the naked eye along an altitudinal gradient from 1500 to 2600 meters. That's where we are going now.
The altitude gradients in mountain areas correspond to marked climatic gradients over short distances, and these are well suited for studying the effects of climate change. These gradients often result in easily identified patterns such as the upper limit of trees and other vegetation, or limits of seasonal and permanent snow cover. However, less obvious changes in temperature, soil moisture, and nutrient levels may also determine the limits and metabolic activity of soil microbial communities. These variations may be of great importance for microorganisms, and therefore to the functioning of the ecosystem as well. The aim of the project Microbes is to carry out a thorough study of soil microbial function and phylogenetic diversity along an altitude gradient of 1200 vertical meters. This is combined with a transplantation experiment where soil is moved from higher to lower altitude and then examined for any changes after 1 and 2 years. The results of these experiments will hopefully improve our understanding of the dynamics and distribution of soil microbial communities. This in turn can help to construct and improve models to predict local and global environmental disturbances in the future.
This perspective, you can see very well the Microbial Observatory, That mountain over there is Monte Perdido. We are in the National Park of Ordesa and Monte Perdido. So the Microbial Observatory starts in that valley that you can see down there. And then we go from that valley, which is located around 1,200 meters. We go up to 1,700 meters, and then we turn left towards that slope over there. And then from that slope, which is around from 1,800 to 2,200, we go up, then we reach a plateau, and from 2,200 we walk up to 2,400, and then from that plateau, then we go towards the third one of the three Marias, which is the end of the Microbial Observatory, which is located at 2,700 meters, something like that.
Soil is an extremely diverse ecosystem, inhabited by hundreds of thousands of microbial species. Just 10 grams of soil can provide a home for as many as 100 billion organisms. That is the same number of cells as there are stars in our galaxy. Still, thanks to technological developments, it's become easier and cheaper to use DNA sequencing to study these complex communities of microorganisms. This has made it possible to identify also the rarer microorganisms and those that cannot survive outside their native habitat. To do this, the scientists in microbes first had to extract DNA directly from the soil samples collected. This is hard work, as the organisms found there often protect themselves behind rigid cell walls that have to be broken down for the DNA to be extracted. The next step is to target or amplify the same gene in all organisms. This is called a marker or barcode, since it helps to identify the organisms. It's done with a technique called PCR, polymerase chain reaction. On a microbial scale, what we think of as soil forms a complex three-dimensional landscape with grains of sand and stone the size of mountain ranges, pores of water acting like rivers and providing the soil matrix nearby with moisture, while other parts may be dry like deserts. The soil is also crossed by plant roots of wildly different sizes that can provide carbohydrates to the microorganisms nearby, in exchange for nutrients like nitrate and phosphates. This complexity also makes it a challenge to study the soil, since no small samples of soil are exactly alike. For this reason, a lot of replicate samples have to be studied in microbes, providing much work for the scientists involved in the field as well as in the laboratory. The Microbial Observatory used in microbes is not the only place where the effects of climate change on mountain ecosystems and their microbial community is being studied. Microbial observatories also exist in the Alps, the Andes, Colorado, Norway and other places. Many of these also take advantage of the new DNA sequencing methods, and the scientists of NEIKER are collaborating with some of them to standardize diagnostic procedures and to see how universal interesting findings can be. Can we see the same changes with altitude in the microbial community here in the Pyrenees as in the Norwegian mountains, for example?
Okay, I am here in Bergen, Norway, to visit Professor Lise Øvros, who has a very similar project going. Let's go.
So my name is Lise Øvros. I'm a professor in geomicrobiology, University of Bergen. And in this project we are collaborating with the plant biologists, where they're looking into the different effects this climate change has on the seed and the plants. And we are interested in similar topics where we're looking into the microbial community, that microbes are the drivers of these biogeochemical processes. So this shows different precipitation rates, and then we can see the composition of the microbial community. It's quite similar, but then we can see significant changes in the area where we have high precipitation rates.
In the microbes project, observations of the structure of the plant community and other parameters are combined with DNA sequencing data covering the 3 domains of the subsurface biosphere: archaea, bacteria, and eukarya. In this way, it provides an integrated approach to improve our understanding of the impacts of climate change. It will also pave the way for the long-term monitoring, which is required to properly understand the interactions between these 3 domains and their changing environments.
We have seen something like 5 or 6 wild boars running into that direction, entering the forest. But we are safe.